Good afternoon and welcome. My name is Ben Choi, and on behalf of my colleagues here at Legacy Venture, thank you for joining this conversation with our esteemed guest of honor, Dr. Francis Collins. For those of you who don't know Legacy Venture, we invest in early stage venture capital firms, and we do that on behalf of a community of philanthropists who commit to use the entirety of their legacy investment for charity. Since COVID began, we've distributed to our investors $860 million, which our community has put to work in their diverse philanthropic pursuits. 860 million is a big number, and yet it seems small in the context of the global health challenges Dr. Collins is fighting at the NIH. I was introduced to Dr. Collins a few years ago as he was interested in bridging the chasm between DC and Silicon Valley. I found him to be incredibly curious, humble, and inspiring. I originally invited Dr. Collins to speak at an in-person event back in early 2020, but the coronavirus took charge and decided that Dr. Collins' attention was needed elsewhere. And it's given me great comfort to know that Dr. Collins has been in the room where it happens when it comes to our national COVID policy decisions. Fun fact, Dr. Collins is a gifted musician and a member of the NIH scientist rock band, the Affordable Rock and Roll Act. Dr. Collins was reappointed by President Biden as the current director of the National Institutes of Health, and he's the only presidentially appointed NIH director to serve more than one administration. You notice he's standing next to his longtime friend and perhaps most famous employee, Dr. Anthony Fauci. Last year, Dr. Fauci was portrayed by Brad Pitt on Saturday Night Live. So, Dr. Collins, I wanted to register my vote for who should play you on SNL. I think Tom Skerritt could do your voice, and he certainly has your mustache. He was awesome as the boss in Top Gun, and so maybe he could pull off playing the boss of the NIH. And if Tom Skerritt isn't available, I'll offer up my own 6th grade son to play you. He already dressed up as you several years ago for Halloween. And there he is, holding that double helix and his guitar. I actually recently learned that Dr. Collins was actually homeschooled by his mother until the 6th grade. And Dr. Collins's post-6th grade accomplishments bring me great hope for my own 6th graders, who were essentially also homeschooled during that first year of COVID. We expect a lively Q&A discussion after Dr. Collins's prepared remarks, so I encourage all of you to please submit your questions at any time through the Q&A button. I know all of you are very familiar with Zoom by now. And with that, I'm beyond excited to welcome Dr. Francis Collins to our virtual stage. Dr. Collins, it's all yours.
My goodness, Ben, what a lovely and surprising introduction. I didn't know those photos and references were going to be part of this, but that was indeed amusing and charming. And your kid is amazing. You sent me that picture of the Halloween performance, and I don't think anybody else has done that, at least as far as they've told me. So yeah, wonderful to see that. And thank you so much for your friendship and for the invitation to come to Legacy Venture today and talk about bridging this gap, as you just called it, between DC and Silicon Valley, a gap which scientifically needs to get closed as quickly as possible because we have so many things that we can do together. And speaking to all of you who are engaged in major ways in philanthropy, there's lots of opportunities there as well for us to work on things together at a time of such enormous scientific promise and scientific need. And certainly COVID-19 has made that clear. I thought in my 30 minutes here, I would be a little personal in my presentation. I mean, I'd give lots of talks about the NIH and what it does, but I'm going to start off talking a bit more about my own trajectory. And I will get at the end into a part of who I am, which is that I'm a person of faith, which surprises some people who think that scientists like me would basically all be either agnostics or atheists, but I'm actually a very serious Christian. And I'll tell you a little bit about that when we get to that point, because it seems to me that science and faith have a lot to say to each other. They have worldviews that are not in conflict, but they're complementary. And it troubles me that somehow in our society, we seem to have adopted a view, at least a lot of people have, that these two things have to stay apart from each other or some kind of explosion will happen. Well, I think what happens instead is wonderful harmony and synergy. So I want to say a little bit about that as we get through some of the science part that I thought I would tell you about to begin with. So I'm going to share my screen so I can show you some images as well as offer you some words. And hopefully this will go seamlessly, because that's what we always want to have happen when you're sharing your screen on Zoom. And as far as I can tell, that sort of worked. And unless somebody's going to jump in and say, we can't see your slide, I assume you can. Okay, I see thumbs up from Ben. So yeah, past, present, and lessons for the future is what I'm here to talk about. So I'm going to turn the clock back a bit and tell you a little bit about my own experience. So I'm trained as an internal medicine doctor, but I'm really excited about the applications of molecular genetics to human diseases. I started out in chemistry. My PhD is in quantum mechanics, which I don't use a lot every day, but it was a great way to think quantitatively about things. So I landed my first job after many years of training at the University of Michigan. And my effort there was to try to see in the 1980s, what could you do to actually track down the genetic cause of a disease when you didn't really know anything about it, except it was inherited? Well, what was that like? It was hard. So I actually got a picture of myself taken in a haystack holding up a needle to just explain why we hadn't succeeded this in the first few months or the first few years. And my big effort was to try to see whether strategies could be developed and applied that would lead to the identification of the cause of cystic fibrosis, one of the most common forms of recessive autosomal diseases in humans, and a very serious one, and one we didn't know much about. And after many years and a lot of efforts to build collaborations with other experts, that did in fact succeed in 1989 with the identification of that gene. And to my great delight, we are now in a circumstance 30 years later where 90% of people with cystic fibrosis, because of this understanding of the gene, now have therapy that really works extremely well. So that was like a big moment, but it was incredibly difficult. And if you were going to do this for the 7,000 genetic diseases where you wanted to find that answer, most of which were going to be even harder to sort out, this was not going to scale well. So we had to have a better way to do this. And the way to do it really was to read out the entire letters of the human DNA code once and for all, have them available. So you, with a click of your mouse, you could pull up information that just didn't exist in the 1980s. Let's read our own DNA instruction book. Well, that was the Human Genome Project. And I was a big fan of it, but didn't really quite expect I was going to be asked to lead it, but I was. And by 1993, left my position at Michigan with a little bit of a lump in my throat because I loved that university, and came to NIH to become, oh my gosh, a federal employee with a responsibility to oversee this very challenging effort to read out 3 billion letters of the human genome. And you have that information inside each of your cells. And to get it really right, and also to give it away, to put that information out there every 24 hours so people could start working with it. I'm not going to go through this long, drawn-out timeline that you see here, but there were a lot of milestones along the way. And we had bumps in the road, and there were times where I thought, what have I decided to do here? I've decided to preside over something that's going to fail. That's not good. But happily, it not only succeeded, by the time we got to 2003, we finished the job ahead of schedule. It was supposed to not get done until 2005. And it took, though, an amazing, talented group of dedicated people to make it so. 2,500 scientists, 20 institutions, 6 countries, 13 years, all of that working together. And here's just one of the photos of the senior leadership of that group getting together. What a dedicated effort. And it was the first big science effort in biology. Certainly we had big science in physics, but we hadn't really done something like this in life science. And the fact that it succeeded, and that it also developed this ethics of you're going to give the data away, right? Because this is not about you. This is about creating a foundation of information that everybody can use. And that's what we were able to do. But of course, it's one thing to have the sequence, those 3 billion letters, written in a language that, well, we really didn't quite know how to read it. 4 letters in its alphabet, A, C, G, and T, 3 billion of those. What does it all mean? And particularly for me as a doc, if you want to use this information to help people, you want to be able to figure out how to help these doctors who look like they're completely bamboozled by the information. How do we turn the genome into functional, life-giving information? That was the next set of questions. So over the next few years, Oh, we funded a whole lot of big projects that would enable decorating the genome with functional information. And every one of these was an incredibly interesting effort, and they all involved big teams, again, working together without worrying who was going to get the credit and putting all the data in public databases. Along the way, we learned a lot of stuff. We learned, for instance, that cancer is really going to be one of the first beneficiaries of this ability to sequence DNA, because cancer is a disease of the genome. If you have a cancer, it's because of a misspelling somewhere in the DNA that's causing a cell to grow when it shouldn't. And if you have cancer, you probably want to know what's wrong with your cells and not just a generic snapshot of somebody else. So cancer genomics became very quickly one of the most major advances that this made possible. And so did making diagnoses in mysterious places, especially in the newborn ICU, where you don't know what you're dealing with. Well, let's just sequence the genome. And amazingly, what cost the first human genome $400 million now costs about $500. All you Silicon Valley people, I would bet that you can't come up with too many technologies that dropped by that number of orders of magnitude in the space of 20 years. And not only is it cheaper, it's faster. You do this in about a day, whereas that first one took us 13 years. And not only is it faster and cheaper, it's better. The quality of the sequence is astoundingly good. So one time in your life here, we have faster, better, and cheaper. Usually it's 2 out of 3. We got 3 out of 3 with all the investments that were made in DNA sequencing. So that was fun. I oversaw a lot of these things. And it was now 2008. And I was getting restless, like, what's next? I don't know. I've been doing this thing with the Genome Institute for 15 years. So I quit and I basically became unemployed. I took a break. They gave me a nice party. They gave me a guitar that I got to design, which has a double helix on the fretboard. That was kind of cool. I got to ride my motorcycle with my wife, Diane, a lot more than I had been because I've been too busy. And I wrote a book about personalized medicine, how you could use DNA and the individual consequences of your own genome to do a better job of influencing your decisions about staying healthy or about how to manage an illness if you develop one, which has become a very big deal. We call that precision medicine now. And it is the way in which, over the course of time, we're getting increasingly away from one-size-fits-all medicine to something that's about the individual, which is both more likely to be successful and more likely to get people's attention in terms of things that they otherwise might choose to ignore. So that was kind of fun. And then a phone call in April of 2009, and it's the president, and he wants me to come back to NIH. So there I am walking into building 1, and I'm still in that role. Appointed initially by Obama, reappointed by Trump, and now reappointed again by Biden. As far as I know, I am the only presidential appointee that has managed to stick it out through more than one administration. And now I'm up to my 3rd president, and it's a privilege indeed to be able to serve in this way. But now my job is not just about the genome and the ways in which it can be used. It's about everything. I'm supposed to know about neuroscience. I'm supposed to know about immunology, which is one of the sciences that always scared me most. I'm supposed to know about rare diseases and common diseases and design of clinical trials. And it's great because it just makes the horizons almost infinite of the kind of science that I can jump into. And I have fantastic people to work with. There are 27 institutes and centers at NIH. They all are run by fantastically gifted, creative, organized scientists. They all report to me, but I don't have to worry too much about them running off in the wrong direction. I need to kind of figure out how to nurture and encourage them, and particularly how to build collaborations between them, because there's always a bit of a tendency for people to do their own thing. And the more you can get a cooperative effort, the more likely it is that something's going to happen. So what NIH does, I think not everybody's familiar with it, even though we are the largest supporter of biomedical research in the world. We have 2 components. We have a basic science part of what we do, the pursuit of fundamental knowledge about how life works, but then the application of that for medical purposes to extend life, reduce illness and disability. And it's about 50-50 in terms of how our budget is distributed. Today, we conduct research in our own laboratories. You see a picture of the NIH campus in Bethesda. That building right there, that's building 50. My lab is right there because I continue to run a lab of about 10 people working on diabetes and on a premature aging condition called progeria. But most of our money is not spent there. It's spent in universities, medical schools across the US, including a lot in California, I might say, who get a lot of our grant monies. And well, they should, because of the creative talent that's out there. So if you hear about a breakthrough in medical research that happened at a US university, it's almost certainly the case that NIH supported that through a grant that the investigator applied for, got funded. We do a lot of training through that. We get the next generation going. We have communication roles, which is a very important part of what we do, especially now. And we think we're pretty good at it by various metrics. You could count Nobel laureates as one, I suppose. 163 of them have been our grantees or our intramural researchers. And just wait till next week, because Nobel week is coming up. On Monday is going to be medicine physiology. Wednesday will be chemistry. I will make a prediction there will be NIH funded grantees in both of those, but of course, I don't know. We'll see. Be kind of surprising if the people who came up with mRNA vaccines don't get mentioned there. That's kind of a pretty big deal, and I'll come to that in a minute. In fact, I'll come to that right now. So having been NIH director since 2009, I had lots of things to think about. And certainly all of us, including Tony Fauci, the remarkable leader of the Infectious Disease Institute, who I speak to several times a day just to try to manage our efforts here, we've all sort of expected the time would come where a pandemic would emerge. We all kind of thought it would be influenza, but after SARS and MERS over the last 20 years, both of which are coronaviruses, we also were concerned that that might be one if it emerged in a way that was even more dangerous, more contagious. And as you know, that's what happened. But it was a good thing that we thought about that. And already there was a pretty vigorous research program in coronaviruses to enable a rapid response. So when this happened, remember this, January 3rd, 2020, what's going on? Pneumonia cases in a seafood market in Wuhan, China. For a few days later, genome sequence of the virus identified. It's a coronavirus. It does have a relationship to SARS and MERS, but it's substantially different. It looks most close to a virus that had been previously derived from a bat in a Chinese cave. We still think, by the way, the most likely origin of this is a natural origin from bats, maybe through an intermediate host to humans. People have also wondered about whether this could have in some way been a lab accident. The evidence against that has really not been there, but it makes good copy, I guess, for people who are writing speculative stories. Most likely, though, this is a virus which has been around in bats, found its way into humans, and turned out to be incredibly effective in both making people sick, but also making them really contagious, and especially really contagious before they had symptoms. That's why SARS-CoV-2, the COVID-19 virus, has been so dangerous, because it is so easy for people to get infected and not know it, and then infect other people. And it spreads so rapidly, especially with the recent variant called Delta. So the big push had to be right away, vaccines. How are we going to develop in record time a way of approaching this very dangerous virus? And here's a diagram of what it looks like. And it was pretty obvious what you want to do is to make a vaccine that will raise antibodies against this spike protein, because that's the part of the virus that your body sees first. And it's also the part that gets the virus inside your cells. So all the current vaccines basically seek to raise antibodies against spike. There are 6 that were invested in as part of Operation Warp Speed back in 2020. But 2 of them, I think, particularly deserve attention, because these are the mRNA vaccines. And we had never had an mRNA vaccine brought all the way through to FDA approval before. But this was built on 25 years of basic science, much of it considered sort of, off in the backwater, like, oh, that'll never go anywhere. Another lesson about science. You need to invest very broadly in foundational basic science, because you never know where the big breakthrough is going to come from. And mRNA, and again, if I'm mentioning Nobels, I would not be surprised if Katalin Karikó, Andrew Weissman, University of Pennsylvania, get mentioned next week as the ones who figured out how you could modify mRNA chemically so that it would not induce an immediate, unfortunate immune response of the wrong kind, and you could instead use it as a vaccine. And we had worked incredibly tightly with them and with others over the preceding 10 years to get to this point. And basically, National Geographic writes about this. It is basically a way to get your own muscle cells to make that spike protein by injecting into them the coding information. That's what messenger RNA is. And having the muscle cells go, oh, this is mRNA. I know what to do with that. I'll run it through my ribosomes and I'll make a protein. And out comes the spike protein, and the immune system goes, whoa, wait a minute. We haven't seen that before. We'd better make an antibody. And that's what these vaccines have done. But very rapidly, with 63 days from when we had the sequence of SARS-CoV-2 and the first patient was getting injected with the candidate vaccine in a phase 1 trial. That usually takes 2 or 3 years, 63 days. And of course, that speed continued. And just 11 months after we learned about this virus, we had 2 vaccines, both of them mRNA, from Pfizer and from Moderna, with FDA providing emergency use authorization and with the efficacy astoundingly high. I have to tell you, Tony and I, thinking about this as the trials were being run and working 24/7 to support that, would have occasional conversations about, well, what do you hope as far as how good this is going to be? None of us ever hoped for 95%. I kind of thought, well, maybe we'll get 60 or 65. And Tony was like, oh, we can get 70. But 95 was totally unexpected and totally wonderful. And those numbers have held up, which is just an astounding thing to be able to say. And certainly, as I'll come to in a bit, as a person of faith, talk about prayer. This was an answer to prayer. Not only did, of course, we need the vaccines, and I'll come back to where we are with those, we also needed to come up with therapies because people were going to get sick. They were getting sick in great numbers, and you don't want to put all of your efforts in a prevention when you have people who need treatment. And for that purpose, I had the privilege of putting together a public-private partnership with all of the parties that I thought could help here, that would enable us to sift through hundreds of possible therapeutic agents and get them quickly into rigorous clinical trials to see what worked. And that is this active partnership, which involves 20 companies, various government leaders, some nonprofits, and very ably managed by the Foundation for NIH. And I serve as the co-chair of the executive committee of Active, along with the chief scientist of Johnson & Johnson. And this is an amazing group who have absolutely thrown themselves 24/7 into trying to do everything possible to prioritize the therapeutics, to set up the clinical trials, to write the master protocols, to evaluate the results. We met this morning for an hour and a half and had a bunch of really interesting revelations to point to. Now, what have we learned from this? Just to quickly summarize, because I could go on for an hour about all the therapeutics that we've tried and what worked and also what didn't work, and you need to know that too. Hydroxychloroquine does not work, in case you still thought it might. Ivermectin might work. We're actually running that trial right now in a rigorous way because none of the previous trials have been adequately designed. But many of the things we've tried kind of have taught us something about what works when. So this diagram is supposed to show you the course of the illness, starting from somebody who has no symptoms but is infected and is contagious, and then milder symptoms, and then some people sick enough to end up in the hospital, initially without oxygen, then requiring oxygen, then high flow, then ventilation, and then sadly, oftentimes death. So what we have learned, and I don't think we entirely knew this before putting this huge effort in here, is if you have an antiviral strategy, like a drug that attacks the viral life cycle, or a monoclonal antibody that attacks the virus directly, those work better early on. They really don't work by the time somebody is super sick. By that time, the immune system is the thing that's really causing the problem. The immune system is overreacting. And so there, you need these immunomodulatory strategies like steroids. And that turns out to be life-saving for a lot of people. Interestingly, this virus also induces blood clotting. So we have learned that blood thinners, anticoagulation strategies, turn out to also be potentially life-saving, but you have to know exactly how much to give and which one to give. And we have learned a lot about that along the way. So we know much better now how to treat people with this disease, all the way from the early symptoms to people in the ICU, but it is still a terribly severe disease, and we are still losing 2,000 people a day to COVID-19, and especially so since this Delta variant appeared. What we really wish we had, and this is a little bit sort of where we're going in the future, would be a very effective, very safe oral agent, a pill that you could take the day you got your positive diagnosis, and it would attack the viral life cycle and you would be done with it in the course of a couple of days. We don't have that. We don't have any drugs that are that precise, but we've learned a lot about SARS-CoV-2, and we think we know how to build that kind of a drug. And so with an additional $3 billion from the Congress, we are deep into this effort to try to develop such drugs, initially for SARS-CoV-2, but probably also for other potential pandemic pathogens that might be lurking out there in the future. Another thing that we focused on very heavily is our concern about health disparities. If you wanted evidence that we have a problem in this country in terms of racial inequities in health access, COVID-19 shows us that in stark and deeply troubling ways. The hospitalization rates and the death rates for African Americans and Latinos are substantially higher than they are for whites. And of course, if we're going to run clinical trials on vaccines or on therapeutics, we want to be sure we're being inclusive in the participants so that we can say whether these things actually work for the highest risk groups. And for that, we started this very aggressively organized community engagement alliance to try to really do the right thing here, reach out to communities to try to involve them in our efforts to explain what is a clinical trial and why would you want your community engaged. And this has been rapid moving and very much put us in a different place than we ever have been to try to consider medical research as a true partnership, not just with a bunch of universities and academics, but with communities that we could enlist as our partners, and partners who really have a place at the table and can tell us when we're doing things that is not going to be well received by their view. SEAL has done a lot in that regard. And then another thing that we've learned about COVID-19, which was not expected and which is very troubling, is that a significant fraction of the people who have this illness do not get better in the space of a couple of weeks like you would expect for a respiratory virus. The so-called long COVID affects as much as a third of people who've had this illness, including people who had fairly mild disease. It's not just the people who are in the ICU. We don't understand this. We don't know what is the pathogenesis. Is it the immune system that gets knocked out of kilter and doesn't get reset? Is there some lingering infection that we can't find somewhere in a reservoir in the body? We don't know. And Recover aims to sort this out by now putting together a cohort of about 30,000 people, some of whom have long COVID already, some of whom are acutely ill, and we're going to try to figure out who gets long COVID and who doesn't. So this is going to be another huge effort, because if you look at the numbers, we may have millions of people already in the United States who are suffering from long COVID, maybe not so much heard from, with brain fog, with confusion about simple matters, with palpitations, with profound fatigue, and we don't know what to tell them to do. So this is another big area of importance. I'm mostly talked about therapeutics and vaccines, but I got to give you a quick snapshot of what we did on diagnostics, because Silicon Valley people, you would have loved all of this. This was technology development in the biggest way. You know that we got off to a bad start in terms of availability of testing for COVID-19 back in early 2020. And the Congress turned to NIH and said, okay, you guys, we need new technologies, including those that can be actually done at the point of care. So you're not sending off a sample to a lab and hoping you get a result back in a few days. So we invested in that in a big way. And we did so in a pretty interesting fashion. We built an innovation funnel, and we invited people who had cool ideas about technologies that might work for doing detection of SARS-CoV-2. And in the space of just a few days, we got hundreds. Ultimately, we got 3,000 applications, mostly small businesses, as you can see here. And then we sifted through them to see which of those had some promise. Well, that was still about 800. And then the ones that looked most promising, we threw them into a shark tank, and we really called it that. And the shark tank had in it business experts, engineers, technology experts, supply chain and manufacturing experts, basically to say, could your technology actually turn out to be useful in the space of a few months, not a few years, a few months. And through that funnel, as you can see, with sort of progressive narrowing with higher and higher bars, we now got to the point we have 35 testing technologies that are out there deployed. When you go to the pharmacy and look on the shelf for home testing, it's because of this program. And people are now able to do their own test at home if they're a little worried about whether they got exposed or have the sniffles, and get your own result in 15 minutes. That is so different than where we were even a year ago. So that's been a learning experience for us. Well, where are we with the pandemic itself, and what lessons have we learned? And here's where there's been a big discordance between all the science that's gone remarkably rapidly and the way in which it's been applied, which hasn't necessarily gone so well. This is the curve of what the number of daily cases looks like in red, starting back at the onset and now going up until yesterday. Yesterday, there were 114,000 new cases, which is pretty frightening. We've gotten over the worst of that most recent Delta peak, but it's coming down kind of slowly. And there were still 2,026 people who died yesterday. The vast majority, over 90% of those, are unvaccinated people. They didn't need to die. That is a terrible tragedy in our ability, or our lack of ability, to convince people to take advantage of something that is safe and effective, namely these vaccines. I didn't see that coming. We have the Delta variant to blame for why things got worse again this summer, because we were kind of doing okay. And then this guy showed up and rapidly, as you can see in the diagram here, just took over the viral population in the space of weeks, and is now 99.9% of all the isolates, because it is so contagious, about 10 times more than its predecessors. So if you're thinking about this, it's not the same pandemic we had last year. It's almost like it's a totally new one, because this guy is so different and it's spreading. Fortunately, the vaccines work, but unfortunately, they're not being utilized in a lot of places. This is what the hotspots look like from yesterday. The Northwest is starting to look bad again. Alaska's having a terrible time. The Appalachian region also, and these are all places where vaccination rates are quite low. The mapping of where the problem is and where people are dying maps quite sadly in the places where vaccine uptake has not been embraced. So what to do about that? Well, this is what vaccine doses look like over the course of the time since they became available, starting January of this year. And we did pretty well there in April and May, and then we kind of hit the wall with people who were not confident, one might say hesitant, and dropped off very quickly. And we have not really seen this come back up, even in the face of all this death and destruction from the Delta variant. We have about 70 million people who are still unvaccinated. They've not gotten their first dose. Every one of those at terrible risk of infection and of spreading it to others. And again, the social media impact here cannot be overstated, because an awful lot of those 70 million people got information from social media that was just frankly wrong, some of it intentionally wrong, spreading rumors, conspiracies that scared people. And once they kind of lock themselves into this mindset of, no, that's not for me, it's really hard to turn the corner on that. Now, let me just finish here with a quick thing about boosters, because that's the topic that's on many people's minds. And then I'll say a little bit about science and faith. This is data coming from Israel, which is where we have the best data right now about what's happening with vaccine effectiveness over time. Basically, Israel has a small enough country with a single vaccine, Pfizer, and they really record everything that happens to everybody. So you know exactly where you are. And this is showing you, let's look at age 60, first of all, what happens in terms of infections in people who are vaccinated. And what's recorded there is the number of months since vaccination. So in Israel, if you were just 2 months away, well, you have that number of infections. But if you were 6 months away, it's clearly higher. So there's a waning of the protective effect. And that happens in all the age groups. Although the people who are 60 and over are more likely to get more significantly ill, certainly illness is happening as breakthroughs occur as the vaccine effectiveness wanes, which is why you want to do something about that, like consider a booster. This one diagram, I think, tells so much of the story here. Just look at the graph on the right. Here, this is Israel again, February through October of this year. They started immunizing right about there. The red line is unvaccinated people. What is their cases? And this is severe disease. This is not the sniffles. This is severe disease, the thing you really want to prevent in people 60 and over. Unvaccinated people, lots of cases. Then they had a pretty nice summer like we did. And then Delta came along. And you can see what's happened. Unvaccinated people in real trouble here. The vaccinated people who got 2 doses of Pfizer, most of them back here in February, March, and April, they were well protected, but you can see things starting to sneak up there with Delta. They're still about 6-fold or 5-fold less in terms of the risk of severe disease than the unvaccinated people. So the vaccine's still working, but not perfectly. What they did is they started boosting their 60 plus population on July 30th, right here. And this green line is what happened to the boosted people. And you can see another 10-fold drop in their severe disease experience. That one diagram tells you why you want to be vaccinated and why, if you're over 60 and it's been more than 6 months since you got vaccinated, you want to boost. And that's basically what has now been recommended in the United States. How to stave off the next pandemic? We need to really prepare for this. We could prepare vaccines. We could prepare drugs against viruses. We have already a pretty good list of the 20 most likely future pandemic pathogens. We should be working on that now and not waiting. Part of what we need to do is to introduce at NIH a high-risk, high-reward culture we're calling ARPA-H. This is the newest potential kid on the NIH block, which I'm pretty excited about because it would enable us to move even more swiftly and take risks in places where we really need to. I won't go through that. But let me finish with just a bit more personal stuff. I started with personal. I'll come back to it now. Yes, I am a person of faith. I was not always so. I didn't grow up in a religious tradition. My parents didn't really think faith was particularly relevant. I became an atheist as a graduate student in chemistry, but as a medical student, encountered up close the circumstances of people who were facing death and realized I would really not know what to do about that, and felt I needed to do a bit more exploration about what was the nature of belief and why do reasonable people apparently sometimes seem to believe. And to my surprise, over about a 2-year period, ended up becoming convinced that God is real, and that after a search through all the various world religions, the person of Jesus Christ emerged as a particularly compelling way that the whole thing made sense. And so I became a Christian at age 27, influenced particularly by the writings of C.S. Lewis, but others as well. And people said, well, your head's going to explode because you're a scientist, and don't you realize that these things aren't compatible? I've always found them totally compatible. I love this pair of images, which sort of reminds people of the beauty of science, because science is beautiful. And you're in awe when you're a scientist and you make a discovery. And if you're a believer, you're also in awe because you just discovered something about creation that gives you a little glimmer of what the creator must be like. So yeah, these two images of a stained glass window and the view of DNA along the long axis, both of them, I would say, are beautiful. And both of them take us to a place of awe. And I feel that every day as a scientist when some new discovery gets made. So I think of science and faith as two ways of knowing. Science tends to answer questions about how things work. Faith has a chance to answer why. Why am I here? I like this Francis Bacon description that God gave us 2 books, both of which mean a great deal to me. The book of God's words, scripture, which I look at every morning, in the book of God's works, nature, and why would those be in conflict? So there's an opportunity here for harmony, and a wonderful harmony that I've found over these last 44 years. And certainly, you could say, faced as we have been with this worldwide pandemic, if there was ever a time to draw on both the tools of science and the tools of faith, this would be it. What's been heartbreaking to me is that those have often, at least in this country, been opposed to each other, which never should have happened. I wrote a book about my own experience called The Language of God and tried to map out the arguments that I have found compelling about how these worldviews are in fact complementary and reinforce each other. And to my amazement, people seem to read it and still seem to be reading it, even though it's now 15 years old. It needs a new edition. Finally, I would say, if you're interested in more of the discussions about science and faith and how they can fit together from people who are really seriously asking questions, but in a civil manner, no mudslinging allowed, go to biologos.org, now led by Dr. Deb Harzma, an astrophysicist trained at MIT, which really invites this kind of discourse to see the harmony, because I think those of us who are in that space find so much to be gained that way and find it so troubling that oftentimes it's portrayed in a fashion that does not reflect the potential complementarity. Biologos.org. So I've kind of romped through a lot of stuff and probably should have cut back the amount of material in order to try to go a little bit more slowly. But I really want to hear your questions. And so I'm going to stop here with this little note from the guy who wrote The Little Prince, because we're all thinking about the future. And I like this. Your task is not to foresee it, because that's really hard, but to enable it. For me as a scientist, as a physician, I want to enable a future where more people have a chance to live out a full, healthy life. As philanthropists, I know you want to enable things to happen without necessarily being quite sure what they will turn into, but aim the arrow in the direction of something that's going to be good for our world. And thank you for all that you're doing to make that so. So I will stop here and see what Ben wants to do with bringing you all into some dialogue here.
Thank you, Dr. Collins. There have been a lot of questions coming through. You've been actually foreseeing some of those questions and answering them along the way. So I'm going to
I've enabled an answer, but I wouldn't be too sure about that.
I will try my best to filter through the ones that are still out there. But please, everyone on the webinar, feel free to use the Q&A button, top or bottom of your screen, depending on what you're on, and we'll do our best to get them in here. Let me start off with a question around variants. Did we get unlucky with the Delta variant, or should we have expected this? And what does that mean for mu and future variants and vaccine resistance?
Oh, great question. No, we weren't unlucky. This was kind of predictable. You have this virus, which although it has a pretty low mutation rate, it has so many opportunities with so many people who are infected with such large numbers of viruses that this was bound to happen. And any virus that ends up being a better competitor than its predecessors is going to spread rapidly. And that's what Delta has done better than Alpha, Beta, or Gamma so far. Mu worried us a bit because it popped up in Colombia, South America, and seemed to be going pretty quick. But it clearly is not as good as Delta in competing in the US. So we've almost not been able to find mu anymore. It kind of got outcompeted. But I do worry about what we haven't seen yet. Is there out there something that has the contagiousness of Delta, the severity of causing really, really serious illness, and is so different in terms of its spike protein that the vaccines that are now still providing really good protection stop working? That is the danger that we'd have to really be considering. And the best way to prevent that from happening is to have fewer people getting infected, because that's where the variants come from. If we have to, we could redesign the vaccine rather quickly, because mRNA allows you to do that. But I hope we don't have to. Right now, we're doing pretty well. The current vaccines are covering Delta, especially after a booster.
Okay, questions around herd immunity. When do we get to herd immunity? Does getting COVID create similar immunity as a vaccine? I mean, probably for the US specifically is the question it's asking.
Yeah, that's another great question. One question is, for the people who have had COVID-19, they have survived, they do have immunity. Is that sufficient? or do you want a vaccine on top of that? The answer is you do want a vaccine on top of that. The natural immunity, although it is a lot better than nothing, it also wanes over time. And there's very strong evidence that a vaccine on top of that will substantially increase your protection. What is herd immunity? Well, it's a little blurry, isn't it? We, at one point, based on other viruses, might have said if you got 75 to 80% of the population immunized, then the virus would basically lose its grip. We're pretty close to that, and the virus shows no sign of losing its grip, partly because breakthrough infections are now happening from people who had natural infection or who are vaccinated. So I will tell you, if we can get to 99%, we'll have herd immunity, but we're still way short of that.
Question on boosters. Given the data from Israel and the supply of vaccines we have, should we not offer booster shots for as much of the population as possible, as soon as possible? If not, why?
Well, the Israel data is compelling for older people. It's not as compelling yet for younger people because they're less likely to get seriously ill. But if you ask me, I think we'll get there. I think we probably will be offering boosters to everybody in the United States as the data builds up over the course of the next month or two. By the way, you may have noticed right now the data is only about Pfizer. Pfizer's the only one that's submitted to FDA for approval for boosters. So people like me who got Moderna, we're going to wait until that data comes through and FDA makes a judgment on it, probably in the next 3 or 4 weeks, and likewise J&J. But ultimately, yeah, I think if we can show that boosters are safe and effective, they reduce the likelihood of illness, and maybe they reduce the likelihood of spreading this further to other vulnerable people, then we should be offering them. And some have been concerned that that's a greedy thing for the US to do when the rest of the world is still waiting, many of them, for their first shot. But actually, it looks as if the US is now in a position to push production. We are offering over 1 billion doses to the rest of the world now. And the amount we would be using for boosters would be substantially less than that. So we can do both. It doesn't have to be either or.
Maybe you've just answered this, but there's a question on international aid. What are the top barriers to distributing vaccines in less wealthy countries? My layman's hypothesis is that vaccine availability is not the top constraint. Refrigeration, corruption, and other factors may be larger barriers. When will our government have fulfilled our duty or self-interest to less wealthy nations?
Oh, well, that's a really good question, because it's not just about do you have the doses, it's can you actually get them into arms in low and middle income countries where there's a lot of logistical challenges. And yeah, the cold chain is part of that, because in many of those places, the idea of keeping something extremely cold for a day or two is just not practical. I think the J&J vaccine is going to help us a lot with this because it has a much more limited requirement for cold chain, and it's a single dose. You don't have to give 2 doses spaced 3 or 4 weeks apart, which is really hard to do also in many parts of the world. We're going to get there, but yeah, the questioner is absolutely right. We got a lot of other issues to deal with if we're going to really have a chance for most of the world's peoples to get access to this vaccine and actually receive the dose.
A question, you were just referencing this, that you got the Moderna vaccine and the data isn't yet there for boosters on Moderna. I won't put you on the spot, but there is a difference between what's best for an individual versus what's best from a public health policy perspective. Maybe an extra booster shot might be right for you individually, but maybe not the best thing for the director of the NIH to set the precedent for. I'm curious, actually, how much of a conflict is there in the case of COVID between individual medical choices that are optimal for one person versus policy for United States, or honestly for the world that you're leading on?
Yeah, it's a great question. And there are people, more than 1 million of them, who've gone ahead and gotten a booster even before it was approved or before their particular vaccine was approved. I'm not one of them. I'm sticking with the rules here. And part of the reason for that is not just, to be waiting your turn. It's also to wait for the data. We haven't yet seen the data, whether somebody like me who got Moderna and then is going to get a booster with Moderna, is there anything there that would surprise you as far as a side effect that we didn't expect from the first 2 doses, and now it's something serious for the 3rd one? So you want to see that evidence. I'm going to argue for that. There's an argument also about maybe you should just switch. So, okay, if they're now giving out Pfizer boosters, maybe I should just run to the drugstore and say, Yeah, I had Pfizer before. Give me one of those. I'm over 65. That would not be true. And again, the mix or match really ought to be tested to see whether that works. If you go from one vaccine to the next one, does that make it better? Does that make it worse? That data is being generated. We'll know that in the matter of actually the next 3 or 4 weeks. And then people can make some decisions. So yeah, individually, maybe people will jump the line a little bit, but if they're doing so on the basis of data that doesn't quite exist. And that's not generally a good idea. Keep in mind, the vaccines are still working really well. It's not like we have a crisis here and everybody has to have a booster tomorrow to be safe. People are doing really well with the vaccines they've already got. The people I worry about, the people who didn't get the vaccine at all, that ought to be our area of greatest focus. And I'm totally frustrated by our inability somehow to get the information in front of those people to convince them to take action.
There are several questions that came in. I think, again, you may have answered this fully, but lots of questions around mixing vaccines. It sounds like, if I heard you right, the data is around the corner within weeks.
Yeah, I think by the end of October, we will know whether mix and match is a good thing or whether you have to stick with the date you brought to the dance and keep the same vaccine on your list.
While we're on timelines, a similar question for approval for children. There's only one, Pfizer's only one right now that's kind of in queue. Where do you think we'll be in terms of that specific ones, but overall its availability for children?
Yeah, Moderna has also submitted data for children. Pfizer's data has been released in a press release anyway, and it looks really good based on more than 2,000 kids aged 5 to 11. They got a really good immune response to 1/3 the vaccine dose. But FDA is going to need to look through that very carefully. Some of us are hoping by Halloween, maybe there'll be an approval, but don't quote me on that because FDA has to do their job and make sure it's safe. These are not just little miniature people, they're kids. We want them to be taken care of in the right way.
You mentioned FDA. One of the questions that came in is Surgeon General, FDA, NIH, CDC. Please explain briefly, like, how does this all fit together?
Well, great question, because we do have a pretty complicated system. We're all part of the Department of Health and Human Services. So I'm the head of NIH. Tony Fauci is in NIH, but he's also the president's advisor on COVID. So he has 2 jobs. The CDC director is at the same level as NIH and FDA as well. And we've worked really hard to try to pull all this together. The Surgeon General, Vivek Murthy, who's a terrific guy, convenes Tony, myself, Rochelle Walensky, Janet Woodcock, the head of FDA, Rachel Levine, who's the Assistant Secretary for Health. And we meet just us at least once a week and try to identify what's a question that really needs an answer that we ought to be sure we know what the answer is. So we're working tightly together, but I get it. It's a lot of people, a lot of mixed in here, but maybe that's not so bad at a time where there's so much happening.
Let me shift gears back to the last topic in your presentation around faith. And it's one of the reasons why I was so excited to have you come speak to this group. You talked about BioLogos and bridging that divide between faith and science communities. And it seems like a major failure to communicate that we have is across that faith line within the US, at least from an identity standpoint, if not an actual belief. And mask vaccines, hesitancy, those things. Where do you see hope for a path out of today's extreme identity politics? What gives you hope there?
You know, it is hard to watch the point we have gotten to, Ben, where everything has become so much polarized and politicized. And the evangelical church, which I'm part of, has certainly gotten heavily affected by that. And when you look at vaccine resistance, for instance, where do you see it? White evangelicals are right near the top of the list because of distrust of science. And this is just heartbreaking. Many of those people, like I, have prayed for answers to COVID-19. And then science, as a gift, I would say, from God, has come forward with those answers, and yet they're being rejected, oftentimes whipped up by information that's provided over the internet that is really, really wrong. And I'm sympathetic with those people who are confused and fearful after all the information that's been thrown at them. I am not sympathetic with the people who are spreading that information, especially if they know that it's wrong. These vaccines do not cause infertility. They are not the mark of the beast, as some are trying to say from Revelation 13. They are not going to give you COVID. They don't have a chip in them that Bill Gates has managed to insert into you if you get injected. A lot of people believe that, and it's outrageously so. And unfortunately, the church has been caught up in this as well. I mean, my hope is that we will see continuing courage of leaders to step forward and learn what the facts are and then spread that around. Pastor Curtis Chang has a whole bunch of videos that really encourage people to watch if you want to see from the perspective of a Christian pastor, what's the truth here? And I've done podcasts with people like Franklin Graham and Rick Warren and Tim Keller trying to spread the truth. And I think those have helped a bit, although they've also generated a lot of nasty email that I get. I don't know, Ben. What I'm worried about our nation is that we have two epidemics. We have the epidemic of a virus called SARS-CoV-2, and we have an epidemic of loss of faith in truth, where opinions seem to carry as much weight as facts. And that is a circumstance that cannot be good for the future of our nation. John chapter 8, verse 32, the truth will make us free. Well, I believe that, but lies will imprison us. And there's a lot of lies out there about COVID that are imprisoning and killing people. This is a circumstance where this culture war is having casualties, not just bruised egos, but people are dying because of our culture wars. We got to wake up to this. I keep thinking that it'll get to the point where people will go, okay, all right, enough. But we've lost almost 700,000 people to this disease. And I would say at least 200,000 of those didn't have to die. And that still hasn't seemed to quite do it. So I'd like to be optimistic. I keep looking for reasons to say we're going to turn the corner, but I'm really having a little trouble.
Oh, wait. Thank you for that. I wish I had a better response. Let me ask you a different question that's come through. The audience here is a group of philanthropists as well as a group from Silicon Valley, technologists, investors in early stage technology companies. The RADx program certainly, I think, caught many of their eye. Hearing about that program, many of them probably participated. I know some of the companies in our portfolio participated. When you put that program together, when your team put that program together in such rapid fashion, in such an intense time, I'm curious how you thought through preventing abuse of the system, it's particularly poignant today because I don't know if you've tracked this story about Theranos, but there's a company, and it's a Silicon Valley company, and they were funded not by traditional VCs, but still, and just it's a diagnostic company, and they went out and fooled a lot of people and potentially hurt a lot of people in the process. Yeah. I'm curious how you thought about that with RADx, and how did you try to protect to avoid the Theranoses of getting in there?
Well, they had to show their stuff, and there was no sort of ability to just gloss over things. If you got thrown in the Shark Tank, which hundreds did, basically you had a week of intense exposure of your technology, including actually showing how it worked in a real-time laboratory setting, and with technology experts who would be quick to pick up on something that was wrong that you tried to gloss over. And most failed in that regard. And then if you got through that one, you went to an even tougher next stage in the funnel with even more expectations. And ultimately, you had to pass muster and get emergency use authorization from FDA. And there's a very tough gate. So I'm quite sure we didn't have people just sort of sneaking through on the basis of data that wasn't there. We had a lot of people fail, and we may have put more people through these stages than we should have if we'd been really demanding right from the beginning. But we didn't want to miss the chance of some rough diamond turning out to be our big hit. And we had a couple of those too. So we basically became a venture capital organization, I guess, but with the appropriate scientific expertise to be sure we knew what was going to work and what wasn't.
Well, we're coming to the end of the hour. Let me try squeezing in one last question. There are several related ones, and this will be hard to answer quickly, but you've done it so far, so I'll try. Yeah, for the philanthropists in our community, they focus on many different areas, not all on public health. But everything's connected to the systems level. And so there are several questions that have come in around social justice, climate crisis, and education reform. And I'm curious how you see the NIH's role touching, affecting any of those things, or those things affecting your work at NIH.
Oh my, in lots of ways. We do have a major program that's scaling up about climate change in health, because we need to be in that space and figure out on a research basis what lessons we could quickly learn. Certainly when it comes to social change and social inequities, that is turning out to be our most significant area of focus beyond COVID-19, because, well, 2020 was a year not just for COVID-19, it was also the death of George Floyd and a variety of other wake-up calls. And we recognize that we NIH are not free of the history here of structural racism, and we need to figure out how to make our workforce more diverse by making it more welcoming, and we need to figure out how our health disparities research agenda can be more action-oriented and not just observing the problem. And we are doing all of those things. So yeah, we're in the middle of that, and we should be.
Well, Dr. Collins, we are at the end of the hour. We, of course, have dozens of questions we didn't get to, but you did an incredible job getting through the ones that we were able to ask. Thank you so much for giving us this precious hour from your day. I don't think it's an understatement to say that all of our lives are literally in your hands. It feels like you've been on this 18-month marathon. And I think of all of us on this webinar like your support team. We're cheering you on. We've got a cup of Gatorade, and we're really hoping that you finish this race really well. You will be in our thoughts and prayers as you lead us out of this and future pandemics.
Well, thank you so much, Ben. Thanks for leading this discussion. And thanks to all the legacy venture people who are doing what you can do to make this world a better place. We depend on you. This whole ecosystem that makes science happen and its applications lead to a better human experience depends on you. And I'm glad to have you as a partner.
Thanks, Dr. Collins. With that, we'll close. This has been an incredible hour. Thank you. And thank you everyone for joining us.
Thank you. Blessings on everybody.